So now in this module, we are going to talk about different mechanisms for transporting proteins into different organelles. So let's look at that. The first mechanism we are going to talk about is gated transport. Protein is trafficked between cytosol and nucleus in this gated transport manner. So because the topologically cytoplasm and nucleus are equivalent, the nucleoplasm is basically continuation of cytoplasm. So the nucleoplasm has specific molecules which cytoplasm doesn't, and vice versa. So these type of gated transport, this occurs between proteins which are being transported inside the nucleus. The nuclear pore, we know that there are pores in the nucleus. We talked about those. These basically nuclear pores, complexes, they function as selective gates. That is why it is called gated transport, that actively transport specific macromolecules and macromolecular assemblies, although they also allow free diffusion of smaller molecules about 5,000 daltons or less. So if a small molecule is something less than 5,000 daltons wants to get into the nucleus, it can get there unhindered. However, larger molecules are selected, and that is basically a function of the nuclear pore which is present on the surface of the nucleus. We'll talk about the structure of nuclear pore later, but let's continue about our discussion about the different mechanisms of protein sorting. The transmembrane transport, membrane-bound protein translocators. So you can think of these membrane-bound protein translocators as special receptors. So when they bind a particular protein, they go through conformational change, and these receptors, like we talked about ion channel receptors, just to clear your understanding, we can think of them as ion channel receptors. So when the protein binds them, the protein that has a specific amino acid sequence, this channel opens up and protein snakes through these translocators and go into the organelle. This mode of mechanism is usually used to transport proteins between topologically distinct organelles. Transported protein molecules usually must unfold to snake through the translocator. Initial transport of selected proteins from cytosol into the ER lumen or cytosol into mitochondria, for example, occurs in this way. However, as you know that protein is, while it is being translated, it is directly injected into ER. However, when we talk about the mitochondria, the protein has already been made and it basically unfolds and then snakes through the translocator. We'll talk about both these processes in more detail later on, but let's look at the third mechanism. Vesicular transport, membrane enclosed transport. Intermediates may be small, spherical transport vesicles or large, irregular shaped organelle fragments ferry proteins from one compartment to another. This is a nice picture which shows the topology of the cell. For example, see, this gray part is basically cytoplasm. So it is basically cytoplasm and nucleoplasm are a sort of continuation of each other. They have many similar molecules. However, some molecules which are present exclusively in nucleus, and there are certain molecules which are present exclusively in the cytoplasm. So, however, we will come back to membrane enclosed transport. Membrane enclosed transport intermediates, as I've said, can be small, and they, for example, transport between ER and Golgi apparatus occurs in this way. Vesicles pinch off the endoplasmic reticulum and they fuse with the Golgi apparatus, and when the vesicles fuse, they empty their cargo in the Golgi apparatus and deliver the protein content or their contents to the Golgi apparatus. So the transport vesicles first have to become loaded with a cargo of molecules derived from the lumen of one compartment. In this case, in our example, it would be ER. From the lumen of one compartment, as they pinch off from its membrane, they discharge their cargo into the second compartment by fusing with that compartment, which I just mentioned. The transfer of soluble proteins from ER to Golgi apparatus, transported proteins do not cross a membrane. So the membranes in this vesicular transport, there is no protein, no entity which is going to cross a membrane. The protein is delivered in a package. That package fuses with the target organelle and empties its contents into the second organelle. So there are two types of sorting signals. Let's talk about those. Sorting signal type 1, it is basically a continuous stretch of 15 to 60 residues. Some sorting signals are removed from the finished proteins. We'll look at some examples of that later on. By specialized signal peptidases, which recognize these sorting signals and they cleave them off and they are not present in the mature protein. For example, a signal peptidase would remove this signal sequence once this protein has been delivered to its particular organelle. Sorting signal type 1 direct proteins from cytosol to ER, mitochondria, chloroplast, and peroxisomes, for example. The sorting signal type 2 is not a continuous segment of amino acids. It consists basically of specific three-dimensional arrangement of atoms on a protein surface that forms when protein folds. In the previous example, the signal sequence was basically a stretch of amino acids. In this case, the sorting signal type 2 is basically distributed at different parts of proteins which are not in continuation with each other. However, when the protein folds, these signals come together, and all these, if you will, the signal patches, they come together and they form the sorting signal type 2. The amino acid residues that comprise the signal patch can be distant from each other in a linear amino acid sequence, and they're generally retained in the finished protein. We talked about that sorting signal type 1 generally is cleaved off the mature protein, but the sorting signal type 2 is not. So here are some sorting signals for your observation. By the way, you're not responsible for learning the sequences of these amino acids and which sequence directs which protein to which compartment. It is just for your knowledge, for you to look at that there are signal sequences that direct specific proteins to specific organelles. That's all I wanted to show. So we have talked about two very important issues: different mechanisms of transport and two different types of signal sequences that direct the proteins to their destination organelle.